Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we'll talk about the auditory system and signal transduction. So let's start with what audition is. Audition is the sense of sound. And for us to hear sound, we need two things: a pressurized sound wave and a hair cell, which is a receptor in the cochlea. Air molecules are pressurized and they try to escape. This makes areas of high and low pressure. These are called sound waves. They can be far apart or close together. Wavelength tells us how close these peaks are and can tell us the frequency also. So if you recall the formula, frequency is equal to wave speed, which is v, divided by wavelength, which is lambda. So frequency and wavelength are inversely proportional. So a higher wavelength leads to a lower frequency, and a lower wavelength leads to a higher frequency. And higher wavelengths or lower frequencies can travel deeper into the cochlea. Think of a wavelength like a stick or a string or something, and the higher wavelength, the farther down it's going to go in. And so how do we listen to different sounds? Well, the ear breaks up frequencies as different frequencies travel to different lengths along the cochlea. So let's talk about the path of sound. Sound first hits the outer part of the ear, this is called the pinna, and then gets funneled into the auditory canal. And here, sound waves hit the tympanic membrane, which is called the eardrum. This is your outer ear. As pressurized waves hit the eardrums, they vibrate back and forth, causing three bones to vibrate. These three bones are in the middle ear, and they're called the malleus, incus, and stapes. The malleus is known as a hammer, the incus is known as the anvil, and the stapes is known as a stirrup. These three bones comprise the middle ear, and an acronym for this is MIS, just with one S though. These are also the three smallest bones in the body, and combined they are referred to as the ossicles. They comprise the middle ear, and the middle ear's function is to amplify vibrations from the eardrum to the oval window. So the stapes is attached to the oval window, which gets vibrated. As it gets vibrated, it pushes fluid to go into the cochlea. All this is in the inner ear, by the way. The cochlea is a coiled, fluid-filled structure. Fluid hits the tip of the cochlea, which is the innermost part of the circle, and makes its way out, but it goes out the round window and pushes out. It doesn't go back to the oval window because there's a membrane in the middle of the cochlea which splits it into an upper and lower membrane. This is called the basilar membrane, and this is upon which the organ of Corti sits. The basilar membrane is the base layer of the organ of Corti, and the hair cells are on the basilar membrane. It also vibrates in response to different sound frequencies. The tectorial membrane sits on top of the basilar membrane and on top of the hair cells. When the basilar membrane vibrates, the hair cells bend on the tectorial membrane, and this stimulates the hair cells. The hair cells are called the hair bundle, and they are made up of little filaments called the kinocilium. The tip of each kinocilium is connected by a tip link, which is attached to a gate of a potassium channel. As the tip links get pushed, this opens the channel, allowing potassium to flow into the cell from the potassium-rich endolymph. Calcium cells get activated when potassium is in the cell, so calcium also enters the cell, and this causes an action potential. The action potential activates a spiral ganglion cell, which activates the auditory nerve. The auditory nerve is actually cranial nerve number 8, and so signal travels via the auditory nerve to the medial geniculate nucleus, which is the MGN, and it's in the thalamus. And then it goes to the primary auditory cortex, which is in the temporal lobe. The medial geniculate nucleus is beside the lateral geniculate nucleus, which we talked about in the visual system, and it serves a similar relay function. A key feature of the auditory system is bilateral processing. Each ear sends signals to both sides of the brain for localization of sound. And so we talked about this earlier, but place theory is what explains how we hear different pitches. It says we hear different pitches because sound waves trigger activity at different places along the cochlea's basilar membrane. And now we can hear frequencies between 20 and 20,000 hertz. The brain uses basilar tuning to differentiate frequencies. Hair cells at the base, which is the start of the cochlea, are activated by high frequencies, and those at the apex are activated by low frequencies. So at the base is at the very start. High frequencies means low wavelength, and you imagine a small string, so it won't go too far. And then at the apex, which is at the very end, you want high wavelength, which would be low frequencies. The region to which sound travels to, the primary auditory cortex, which is in the temporal lobe, is also separated by regions to detect different frequencies. This is how the brain can distinguish between different sound frequencies, and this is called tonotopical mapping. That's it for today. We went pretty fast over a bunch of concepts, so feel free to rewatch. Next time we'll go over the vestibular system.